grade 10 students, welcome to our virtual class for the third quarter, week 4. I'm Abigail Sarmiento, your science teacher. Our lesson for today is about heredity, inheritance, and variation. Introduction. We learned in grade 9 that many genes in plants and animals behave differently than genes that Mendel studied, where traits are not entirely controlled by dominant and recessive genes. We also learned how the genes in their deoxyribonucleic acid or DNA influence their characteristics. Now you will work on activities to assess your understanding on the structure of the DNA. Explain how DNA replication takes place, how ribonucleic acid or RNA is made using the information from DNA, how information in some genes is translated into protein, and explain how mutation may cause changes and function of a protein. These investigations on how the genes control cells were done even before scientists first knew that genes were made of DNA. The American genetics, George, Bell, and Edward Patton, established the connection between genes and enzymes. The experiment on Bell and Patton used genes to handle products of cells and showed the importance of genes in cellular activity. In other words, a gene is a portion of DNA that contains the instructions for the synthesis of specific RNA or protein. Building a house usually requires a blueprint, or a plan of the structure of the house to determine how it would look like after construction. Organisms have blueprints which contain information that will determine their physical and chemical characteristics. This blueprint is a DNA is a double helix molecule composed of complementary strands of deoxyribonucleic units. The complementary base pairs of the DNA are held by hydrogen bonds. RNA is a single-stranded. Examples of RNA types include mRNA, rRNA, and tRNA. In DNA, adenine always bonds with thymine, and cytosine bonds with guanine. In RNA, adenine bonds with uracil, and cytosine bonds with guanine. Application. In 1953, James Watson and Francis Crick worked out the DNA is a double helix stranded system. The two sugar phosphate backbones make up the sides of the base pairs make up the branch or steps of the twisted chain. Deoxyribonucleic acid have been during interphase prior to mitosis and meiosis. It is important that new copies are exactly like the original copies. The structure of the DNA provides a mechanism for making accurate copies of the molecule. The process of making copies of DNA is called replication. When DNA replicates, two identical copies of DNA molecules are produced, which are exactly the same as the original. The central dogma of the transfer of genetic information is outlined below. See the picture and observe. What do you think is the role of RNA in making proteins in the cell? Can you imagine a car being assembled in a car factory? By way of analogy, different cars are being built in many simple steps. Engineers tell workers how to make cars, and the workers follow directions to build the cars. Suppliers bring parts to the factory so they can be installed in the car. Protein production is similar to car construction. It is the role of the DNA to provide workers with the instructions for making the proteins, and the workers build the proteins. Other workers, known as amino acids, bring part to the factory. RNA molecules, or the workers for protein synthesis, get the instruction from the DNA on how the protein should be assembled. Moreover, do you know that there are 3 types of RNA that help build proteins? You can mRNA or the tRNA supplies amino acids to the ribosome to be assembled as protein. Translation. The DNA directs the production of proteins and determines the formation of mRNA. The order of bases of mRNA determines the protein synthesis. In translation, each set of 3 nucleotides in an mRNA molecule holds 4 1 amino acids in a protein. This explains why each set of 3 nucleotides in the mRNA is called a codon. Each codon specifies a particular amino acid. For example, the first codon is arginine-uracil-guanine. Instruct the ribosome to put the amino acid ARJ or arginine in the protein. The sequence of the codons in the mRNA determines the sequence of amino acids in the protein. 1. As the translation begins, mRNA binds to a ribosome. Then, tRNA molecules, each carrying a specific amino acid, approach the ribosome. The tRNA anticodon pairs to the first mRNA, start codon, RNA, uracil, and to form the initiation complex. The 2 molecules temporarily join together. Step 2. Usually, the first codon on the mRNA is AUG, which codes for the amino acid synthesis. AUG signals the start of protein synthesis. Then, the ribosome slides along the mRNA to the next codon. Step 3. A new tRNA molecule carrying an amino acid pairs with the second mRNA codon. Step 4. When the first and second amino acids are in place, an enzyme joins them by forming a peptide bond between them. Step 5. As the process continues, a chain of amino acids is formed until the ribosome reaches a top codon. Example, UAA, UHG, or UGA. On the mRNA strand, the polypeptide chain is released. Protein synthesis is occur in gamete cells. Gene mutation is a permanent change in the DNA sequence that makes up a gene. Chromosomal mutation occurs at the chromosome level, resulting in gene deletion, duplication, or rearrangement that may occur during the cell cycle and meiosis. It may cause by parts of chromosomes breaking off or rejoining incorrectly. Most mutations are harmful. Some mutations in a body cell are known to cause cancer, while mutations in insect cells can cause birth defects. A severe mutation may lead to cell death and may have no effect on the body. Sometimes mutation may be useful for the species. For example, a mutation in blood proteins prevents viruses or parasites to thrive in host organ tract. When is mutation inherited? Why are mutations in infected cells heritable? Many diseases are caused by the effect of inherited genes. In most cases, there is only a small difference between the inherited defects in the defective gene and normal one. This difference is enough to cause acute and often fatal diseases. These disease-causing genes are the result of DNA mutation. They may be passed from one generation to the next if present in gametes. The figure shows changes in the single cell nucleus of normal hemoglobin and the one affected by mutation. A recessive gene causes sickle cell anemia, where most of the red blood cells stiffen and become sickle-shaped infected people. This disease cells carry less oxygen than normal cells. People affected by the disease eventually die. Consider what might happen if an incorrect amino acid is inserted in a growing protein chain during the process of translation. Do you think this will affect the structure of an entire molecule? This can possibly happen in point mutation, where a change in a single base pair occurs. Read the two sentences below. What happens when a single letter in the first sentence is changed? What was deleted? Would the result be the same if there would be an addition of a single base? Mutation in chromosomes may occur in a variety of ways. Sometimes parts of the chromosomes are broken off, lost abnormalities in chromosomal structure may occur during meiosis. The normal process of crossing over and during deletion may be affected such that chromosomes break and reunite the wrong sex. If there is a loss or gain of chromosomal material, there can be significant clinical incidence. Changes that affect the structure of chromosomes can cause problems with growth, development, and function of the body systems. These changes can affect many genes along the chromosome and disrupt the protein chain connection. Structural changes can occur during the formation of egg or sperm cells in fetal development, or in any cell after birth. Pieces of DNA can be rearranged within one chromosome or transferred to two or more chromosomes. The effects of structural changes depend on their size and location, and whether any genetic materials is gained or lost. Some changes cause medical problems, while others may have no effect on a person's health. The gain or loss of chromosome material can lead to a variety of genetic disorders. Human examples are the following. First is we have reproductive. It's caused by the deletion of part of the short arm of chromosome 5. Reproductive is French, and the condition is so named because affected babies make high-pitched cries that sound like a cat. Affected individuals have wide set eyes, a small head and jaw, are moderately to severely mentally retarded, and very short. The next one is Down syndrome is usually caused by an extra copy of chromosome 21 or trisomy 21. Characteristics include decreased muscle tone, stockier build, a asymmetrical skull, slumping eyes, and mild to moderate mental retardation. Next, Edwards syndrome, which is the second most common trisomy after Down syndrome, is a trisomy of chromosome 18. Symptoms include mental and motor retardation and numerous congenital anomalies causing serious health problems. About 99% die in infancy. However, those who live past their first birthday usually are quite healthy thereafter. They have a characteristic and appearance with clenched hands and overlapping fingers. Next is we have Jacobsen syndrome, is also called terminal 11th deletion disorder. This is a very rare disorder. Those affected have normal intelligence or mild mental retardation with poor or excessive language skills. Most have a bleeding disorder called ribosome syndrome. Next is we have Klinefelter syndrome or XXY. Men with this condition are usually sterile and tend to have longer arms and legs and be taller than their peers. They are often shy and quiet and have a higher incidence of speech delay. The last one is we have Turner syndrome, X instead of XX or XY. Female sexual characteristics are present but underdeveloped. They often have a short stature, low hairline, abnormal eye features and bone development, and a cave-in appearance to the chest. Human karyotyping. Occasionally, chromosomal material is lost or rearranged during the formation of gametes or during cell division of the early embryo. Such changes, primarily the result of non-disjunction or translocation, are so severe that the pregnancy ends in miscarriage, meaning loss of an embryo or fetus before the 20th week of pregnancy, or fertilization does not occur at all. It is estimated that 1 in 156 live births has some kind of chromosomal abnormality. Some of the abnormalities associated with chromosome structure and number can be detected by a test called a karyotype. A karyotype is an image of the full set of chromosomes of an individual that displays the normal number, size, and shape. Karyotypes may reveal the gender of a fetus or test for the certain defects through examination of cells from uterine fluid, a procedure called amniocentesis, or through sampling of placental membranes as shown in the picture. Chromosomes commonly derived from actively divided white blood cells are stained and photographed. The homologous pairs of chromosomes are identified and arranged in order by size. With the exception of the sex chromosomes, these appear last as shown in Figure 12. These tests are typically done in a blood sample, although any body cell could be used. The cell must be undergoing mitosis, so that the chromosomes are replicated, condensed, and visible under the recombinant DNA. This process uses restriction enzymes to cleave one organism's DNA into fragments and other enzymes to splice the DNA fragment into a plasmid or viral DNA. Transgenic organisms are able to manufacture genetic products foreign to them using recombinant DNA. Genetic engineering has already been applied to bacteria, plants, and animals. These organisms are engineered to be of use to humans. Figure 13 shows the method for producing recombinant DNA. That is it for today's